The overall goal of this procedure is to mimic ischemia reperfusion in C. elegans. This is accomplished by moving C. elegans to a microtubule and washing the bacteria out. The second step is to create an anoxic environment using a lab-made hypoxic chamber. The animals are subjected to anoxia for 20 hours and then placed in aerobic conditions. After 24 hours, the final step is to score for dead animals and score for touch response. Neuronal modifications are also observed through fluorescence microscopy. Ultimately, Anoxia-starvation in C. elegans is used to create the same conditions seen in ischemia injury in superior eukaryotes in an easier and more flexible genetic model.
And the main advantages of these techniques over existing methods of ischemia reperfusion in cells and isolated hearts is that C. elegans is a very powerful genetic model and also is a transparent organism, allowing you to use several different fluorescence techniques. In addition of that, we are showing you how to, instead of using an expensive hypoxia or environmental chamber, to use a lab-made hypoxia and environmental chamber that will just cost a few bucks. Generally, individuals new to this method need to be aware of some key points, as for example, purging the buffers and being very careful with scoring the worms, and also being constant with the amount of temperature that is being used and the amount of oxygen. All of these points can change the outcome of the experiment.
To prepare a custom hypoxic chamber, a Tupperware style